So today our group will be presenting about new treatments for acute lymphoblastic leukemia. An overview of what we will be talking about is a background on acute lymphoblastic leukemia, what are the causes, chemotherapy, target therapy drugs, stem cell transplantation, CAR T cells, background on that, and benefits and side effects of CAR T cells for ALL patients. So, what is acute lymphoblastic leukemia? So it can be called ALL, which is a type of cancer that is most commonly found in children. ALL is a rapid proliferation of immature B and T lymphoblasts and originate in the softer inner portions of bones known as bone marrow. And bone marrow contains stem cells that develop into different types of blood cells, which function to protect and strengthen our immunity from diseases and different types of cancer. There are two subtypes of ALL, known as B-ALL and T-ALL, which differ with regards to treatment of the disease. Known and proposed causes, typically hematopoietic stem cells give rise to lymphoid cells which differentiate into B and T cells, but a translocation between chromosome 9 and 22, known as Philadelphia chromosome, can restrict hemopoietic stem cells from functioning properly. Another proposed cause of ALL is aneuploidy, which is the deletion of transcription factors that are necessary for differentiating of progenitor cells. The Grieve hypothesis results from a weak immune system which stimulates ALL. And when the immune system is overstimulated, it can cause mutations in pre-initiated cells, resulting in B and T cells. And these B and T cells outnumber the normal blood cells in the body. Exposure to chemicals such as car emissions that contain topoisomerase II poisons and deficiency of purine nucleoside phosphorylase are also known as a cause of ALL. Chemotherapy remains the leading treatment for nearly all cases of ALL. Traditional and new combinations of chemodrugs are constantly being developed and tested. Some of the new chemodrugs being used are chlorphorabine and nelarabine.
Okay, so one of them, like Sabrina said, was chlorphorabine, which is also known as CIF. CIF works to specifically target high proliferating cells, such as T and B lymphoblasts. So CIF first enters the cell via simple or facilitated diffusion. And once it enters the cell, it is phosphorylated by deoxycytidine, MP, and DP kinases, which forms its activated form CIF-TP. And so once activated, it has two functions. One function is to allosterically inhibit human ribonucleotide reductase, also known as HRNR. And this prevents the production of NTP to DNTP. And so these low levels of DNTP allow CIF-TP to compete with it into the incorporation of DNA. And so once the CIF-MP is incorporated into the DNA via DNA polymerase, DNA elongation is terminated, which is quickly followed by apoptosis. Okay, and then the second therapeutic drug is also known as nelarabine. And so high levels of DGTP are known to cause originate or give rise to T lymphoblasts. And so nelarabine is a different form of ARAG, which specifically targets these T lymphoblasts. It is just more soluble so that it can be diffused or inserted into your bloodstream. And so before entering the cell, nelarabine is converted to ARAG via the enzyme adenosine deaminase. And so once converted, it diffuses through the nucleoside transporters across the membrane. And so once in the cell, like the other chemotherapeutic drug, it is also phosphorylated. However, it's phosphorylated by different enzymes, those being deoxyguanosine and deoxycytidine. there's ones it shares in common with. But this yields ARAGTP, and this is the activated form. And so once activated, it competes with the deoxyguanosine triphosphate, the GTP, to be incorporated into the DNA. And so once incorporated, like the other chemotherapeutic drug, apoptosis occurs quickly after. Okay, so there's another treatment for ALL. This is known as like a target cell therapy drug, and these specifically target gene changes found in ALL cells. So a common gene change that yields ALL cells is the Philadelphia chromosome. And so like Sabrina was saying, it's caused by a translocation between chromosomes 9 and 22. And so there's a head to tail fusion of the ABL gene from chromosome 9 to the breakpoint cluster region of BCR on chromosome 22. And so this mutation ultimately yields 210 kDa and 190 kDa proteins. And so each is unique to a different disease. So the one that is unique to B, acute lymphoblastic leukemia, is the 190 kDa, and it is seen in 2/3 of it. So it's relatively common. And so these proteins yield a BCR albilsin tyrosine kinase, which activates a large number of signal transduction pathways, and this can lead to resistance in apoptosis, growth factor independence, and alteration in cell-to-cell or cell-to-cell or cell-to-matrix interaction. And these are all factors of a cancerous cell. And so to prevent this from occurring, in the next slide, so there's a specific drug that targets Philadelphia chromosome positive ALL, and this is known as imatinib. And so imatinib acts as a BCR ABL inhibitor, and it specifically binds to the ATP binding pocket of the BCR ABL kinase. As you can see on the left-hand side, ATP is bound, and so this results in many signal transduction pathways that result in ALL. But since on the right-hand side, imatinib is bound to the ATP binding pocket, which makes it so ATP can no longer bind, and so this results in an inactive BCR ABL kinase, which inhibits signal transduction pathways. There's no activation there, and this prevents ALL. And so there was an interesting study in 2026 where imatinib was used on PH plus ALL patients, and results showed a complete remission of 96%. And this is huge. And there was other studies that kind of had the same exact percentage. However, they also followed the same trend of relapse. And this was because of the imatinib resistance. And so on the next slide, the resistance is due to a point mutation in the BCR ABL complex. And so this leads to a decrease in affinity between the imatinib and the ATP binding site. And so this basically makes it so that it's no longer an effective tool at suppressing the BCR ABL activity. So there's going to be some ALL that's going to be produced. And so there are second generation inhibitors that can prevent this from, that specifically target the imatinib resistant variants of the BCR ABL kinases. However, usually a point mutation means that the cancer is far along, it is a progressed form, so you can only do so much in this case. Okay. And then, so there's another treatment with regards to ALL. This is known as stem cell transplantation. And so in this case, so the intention is to destroy cancer cells that have formed in the bone marrow of ALL. And so we want to destroy these and then restore it with new healthy blood-forming stem cells. And so as you can see in the allogenic transplantation, you use donor cells, and this can be kind of dangerous. Usually this treatment is used as a last resort because some major side effects can be transplant rejection, which is when the immune cells attack the patient's own body, and this is very dangerous. And so that can result from the allogenic transplantation. And like, long story short, the normal T cells from the donor are processed and isolated and frozen, and then the ALL patient is dosed with high amounts of chemotherapy to decrease the cancer cells and like basically eliminate them, and then they inject the healthy T cells into the patient in hopes of a recovery and no cancer. And so for the autologous transplantation, they're not using a donor, they're just using the same patient. They try to find a healthy T cell and they try to produce more of them and then give them high dosages of chemotherapy and kind of do the same thing. However, this isn't the most efficient process, and usually there's a relapse afterwards. Okay.
All right, so T-cell immunotherapy is another type of treatment to treat patients with ALL. This treatment uses T cells to fight off cancer. There are two types of T cell. So the first one is the killer T cell or a cytotoxic T cell. This type of T cell will have receptors specifically to bind to the antigen expressed on the infected cell. This type of interaction is an example of the lock and key model. So once this killer T cell recognizes the infected cells, it will release perforin and cytotoxin to kill the infected cells. Once the killer T cell destroys the infected cells, the helper T cell will come and make a chemical signal so that the killer T cell can copy itself and fight off the cancer. Once this infected cell is destroyed, it will be fragmented, and these fragments will be ingested by macrophages.
Okay, so a specific type of this T cell immunotherapy is called adoptive cell transfer act, also known as ACT. And so basically this method uses the patient's own T cells to target leukemia cells such as B lymphoblasts. And so the most reliable method in this case is the CAR cell immunotherapy. And so CAR stands for chimeric antigen receptor. And so it's basically a synthesized receptor that is specific to the antigens on the surface of leukemia cells. For B lymphoblasts, they specifically have antigens CD19 and CD22. And so once genetically engineered with an autologous T cell from the patient, it yields CAR cells which then attack the cancer. And as you can see on the right-hand side, there's a whole process of how this happens. And so basically, long story short, they take the T cells from the patient and then they bring it to a lab and they genetically engineer it using an inactive virus, which inserts the genes into the T cells, and then the T cells express this gene via transcription and translation, and it has these receptors that specifically target the antigen on the cancer cell. And so the patient is dosed with a high dosage of chemotherapy, and then the CAR cell is introduced to that patient and targets those cancer cells. Okay, so on the next slide, we have the CD19 CARs, which are specifically unique to neoplastic and normal B cells. And so the specific treatment is called Chimera, and so it has a chimeric antigen receptor that specifically binds to CD19. It's called CTL019, and so it is synthesized with the T cell and yields the CTL019 T cells, and so that basically targets the cells that express the CD19 antigen and destroys them. Okay, so there can be some resistance. So in some cases, some B lymphoblasts are able to adapt to this type of therapy and will actually lose their CD19 receptors. So therefore, these cells can no longer target them. And actually, it's a very prevalent condition, 50% of patients treated with CD19 therapy actually go into relapse. So, but this is not a problem because there are other antigens on the normal and neoplastic B cell surface, and so one of these being CD22. And so CD22 CARs work in the same way as the CD19 CARs and they work to target and eliminate B lymphoblasts. Okay.
All right, so this treatment can have some side effects, which includes B cell aplasia, which is the mass die-off of B cells. This happens because B cells also express the antigen CD19 on their surface, and as CAR T cells are engineered or modified to target these antigens, they will target the B cells without differentiating infected cells and healthy cells. Cytokine release syndrome, or CRS, can also be a side effect. This happens because CAR T cells will release cytokine as they multiply. This can lead to high fever and low blood pressure. An example of a cytokine release is IL-6, which is secreted by T cells and macrophages in response to inflammation. This side effect can be a marker for the treatment being successful. So another more fatal side effect can be cerebral edema, which can lead to brain swelling and nerve impairment. Another method for CAR T cell therapy is the use of nanoparticles, which will be used to modify the genome of the patient's T cell from inside the body to express CARs. It can have many advantages, like it's not unique to each patient. It is also less expensive, and it is an off-the-shelf treatment. Although this type of treatment has many advantages, doctors or scientists cannot control how many CAR T cells are produced, and this may lead to severe CRS. And this is an example of a nanoparticles used. They have some key components that can be the gene that codes for CARs to target the specific antigen. This is found on the DNA of the nanoparticle, which we can see on the right in white. And then we have nuclear localization signals and microtubule associated sequences, which will help the gene for CARs get inserted into the T cell's DNA, and we can see them in red. And lastly, we have proteins added on the nanoparticle surface to bind selectively to T cells. These are shown on the right in yellow.
So overall, proper DNA replication and repair is vital for development, growth, and tumor-free cells in all multicellular organisms. Unfortunately, some of these factors we can't control, such as the rate of cell division, which can cause crucial health issues that are difficult to overcome. Cancer is a vigorous disease and remains difficult to find the proper treatment, but emerging therapies are displaying positive effects for the future. These different types of treatments are constantly being researched and tested in hopes of a cure for cancer. And this concludes our presentation.